Good afternoon, or good morning, actually. It's now my pleasure to introduce our next fireside chat guest, Dr. Bruce Goldsmith, President and CEO of Passage Bio, and Alex Patopoulos, Chief Technical Officer at Passage. So this fireside chat will be about 25 minutes, and if anyone in the audience has any questions during the session, feel free to type them into the question box underneath the video player, and we'll try to ask them on your behalf. So thank you again both for participating today, and I'd like if you could start off with providing a brief overview of Passage and its manufacturing capabilities for audience members who might be new to the story.
Hi, Gyula. Thank you very much for hosting us, and thank you for Chardan for inviting us. Alex and I are very pleased to be here on behalf of Passage Bio. So I'll give a brief overview, and then I'll turn it over to Alex. So Passage Bio, as you may know, is focused on bringing transformative medicines to patients, specifically for patients with devastating CNS disorders. We were founded in partnership with Jim Wilson's gene therapy program at the University of Pennsylvania. And in partnership with him, we have access to a total of 17 potential CNS gene therapy programs through that partnership. We have 3 programs now in the clinical stage, and we are filing an IND for our 4th program. And in that light, the manufacturing and CMC strategies and the capabilities are really integrated with the clinical strategy as we've executed, as well as the forward-looking commercial strategy of the company. And in addition to the clinical programs, we have an extensive pipeline at various stages of preclinical stages that also require CMC capabilities to enable them and bring them to patients. So with that as kind of an overview and the framework, I'll ask Alex maybe to add a little bit more specifically about the manufacturing capabilities.
Thank you, Bruce, and thank you for having us on the summit. I would say from the beginning, from the early stages of the company, we made, as Bruce indicated, a significant investment in capabilities. And if I can summarize those capabilities for the audience, I would say we have a very flexible, scalable manufacturing platform that can help us advance our pipeline. We have dedicated manufacturing capacity, so we control a lot of the capacity for the manufacture of these complex products. We have established an end-to-end supply chain, starting from the early raw materials like plasmids, all the way reaching the patients across the globe. We are conducting for all our programs clinical trials globally. And that requires to establish a very complex supply chain in order to meet requirements, not only US, but also ex-US. We have established our internal capabilities from analytical and process development perspective. That was a significant investment that we made last year. And we continue to make more investments in that area that give us more flexibility and strength to take control a lot of the complex analytics and the complex manufacturing of these vectors. Last but not least, we have built a significant CMC team with able to not only to operate the labs or oversee the manufacturing, but also to conduct a lot of interactions with regulatory bodies across the globe. So regulatory CMC is one of the strengths that we have as well.
Great. So maybe to start off, in terms of the interaction and relationship with the gene therapy program at Penn, can you expand on how that relates to manufacturing and what that means for Passage's strategy?
I'll take this one, and maybe Bruce can add a couple points. But first and foremost, we have a very strong and deep relationship with Penn's GTP program, and within the CMC, with also the vector core group within the GTP program. We have access to next generation capsids. We're working on interesting payloads for both gene transfer and gene regulation. We have access to next generation analytics, and also we have access to next generation manufacturing. What we have tried to do is really take that strength, the R&D strength from GTP, and integrate it with the capabilities that we have built and we continue to build at Passage Bio. We have a very strong, if you want, discovery and research engine at Penn's GTP, and what we try is to link and integrate that with a very strong CMC development engine for both late-stage development and commercialization. We have integrated technologies, and actually at this stage, based on a lot of the capabilities we have developed, we have a lot of interesting interactions both ways. So we are looking into how we can make more efficient the transfer of candidates as they get through the candidate selection in the research to move very quickly into the clinical development.
And Gila, maybe just to add one thing to that. So the way we think about this is kind of a seamless interaction and project by project and technology by technology discussion with University of Pennsylvania's team, which is really fantastic. And so over time, when I think Alex and I were here a little over 2 years ago, there were very few people in the CMNC team and just an emerging regulatory CMNC team. Now that we have established a much larger team and the dedicated suite at Catalent, et cetera, and the analytics, it's more of a partnership and more of a seamless transition. So each piece of either internal or external or partnership with Penn or we're driving it ourselves is kind of discussed and agreed upon between the two groups. And that's what makes it such a powerful collaboration.
Mm-hmm. Great. And so in terms of those different capacities, the internal versus external, can you expand a little bit on how you've gone about deciding which elements of manufacturing or analytics to internalize versus which to advance together with partners?
Sure, I can start it again. Bruce can add a couple points here. For one thing, if you look into from the analytical side, for example, we try to internalize the most critical assays like droplet digital PCR, in vitro potency assays that we are looking into for full empties or for characterization of the capsids, that they are very complex tests. Where externally we maintain more the standardized compendial or assays that we can keep with external partners, and then also with the CMOs, the CMO we're using for the dedicated capacity. There are certain assays they stay in order to facilitate the manufacturing with the CMO. The same way with the manufacturing, the key elements that we have tried to take internal is more or less the testing, as is associated with the analytical and the QC testing. As we advance our capabilities, our internal analytical and QC capabilities, but also we maintain right now a very strong relationship, in this particular case with Catalent, having a dedicated capacity because they have also all the GMP systems in place. So we are trying to maintain a very balanced approach in our sort of what we internalize, what we externalize, keep external. And then we are looking into, as we advance the programs, what makes more sense to internalize gradually and in favor of the program that we're trying to advance.
Got it. And in terms of scalability and capacity, is your current manufacturing plan sufficient to support what would be needed for a launch at commercial scale? And if not, what is your strategy to get to that point?
So we have a large pipeline, but at the same time, we have not only the dedicated suites at Catalent, the dedicated suite at Catalent, but we have also a platform, manufacturing platform that operates even at the clinical stage right now on the commercial scale. So to that end, based on what we know today for these programs, we feel comfortable and we feel confident that we can launch some of these programs. As we advance and as we get more experience, I need to remind them that we are still early in the clinical development for a number of these assets, and we are setting dose levels. We are looking very carefully how much we expand externally, or how much, or how we are going to, proceed with a hybrid model when we need to internalize some of the manufacturing capacity.
Got it. And let's touch on the manufacturing platform specifically that you're using HEK 293 transient transfection system. What do you see as the advantages here? And also maybe what innovations in manufacturing are you looking at that could help the program kind of get to the next level in terms of exceeding goals that you're setting for yourself now?
It is true that there are a lot of different or a number of different manufacturing platforms that a number of in vivo gene therapy companies are using. However, when you look into those platforms, you need to see them in the context of the dose level, the patient populations you are targeting, the pipeline that you are managing with, and the overall strategy from drug development. And those considerations, they go into selecting or making a choice on the platform. As you pointed out, it's very true. We have made the commitment, and we are committed in the mammalian production platform for vectors using the transient HEK 293 platform. The platform for us it's for one thing, it has the benefit that it's very flexible. It's a plug and play platform that we can easily change products, change serotypes, change payloads, and really manage efficiently all the manufacturing capabilities, the testing capabilities as they pertain to the programs we advance. It has a regulatory acceptance and provide us with the ability to integrate also our drug development, as I pointed earlier, with PEN, and how we can quickly move these candidates from research to clinical development. Considering the fact that we're using ICM for our delivery and the dose levels, at least we are looking into the clinic. As I pointed out, this platform we believe can get us into the early stage of commercialization and launch on some of the programs that we are, some of the diseases that we're trying to address.
I guess one other comment on that is that 3 of the programs, 2 in the clinic and the IND we're going to file, are all using the AAV HU68 capsid as well. So, Alex and his team have been working really diligently to advance not only the analytics, but also process development and efficiency across those 3. So that also adds to kind of the platform capabilities that we have.
You ask about some innovations. I want to point out it's very hard to point out 1 or 2 innovations because we are talking about here an emerging modality, an emerging platform. And to that end, the innovations are happening across sort of the different parts of drug development and manufacturing. If I have to select a few things, just to give an indication, some of the technologies that they're impacting or they're changing the way we manufacture and test the vectors, I will start with the analytics. We are heavily using more and more NGS in order to characterize the vectors. We are on the manufacturing side, we are looking into ways how we can improve yield and robustness of the process, like in the transfection phase of the HEK 293s, and also even on the design of the payloads and the plasmids and how all of those things, they are coming together in order to provide a robust, high quality product at the end of the production, the manufacturing.
Got it. And are there CMC challenges, or what do you see as the biggest CMC challenges with regard to either implementing a new innovation or just in general within the space that you've been thinking about?
Well, there are a number of considerations here. I would start first with, if you want, with the resources. It takes a lot of resources to implement and a lot of these new technologies. We are looking into very carefully how we're trying to introduce some of those things in a way that we are not introducing major bottlenecks in our clinical development and also in fact require significant resources to implement them, but more of a staged or a staggered approach and very careful, but also the consideration with risk from a regulatory perspective. We are very careful how these changes are being introduced, looking more how we move some of these innovations from one product from one stage to the other, or from one product to the next product. So overall, the consideration, the challenges, I would say, they may be the financial aspects of those things and the regulatory aspects.
Got it. So we talked about data analytics a little bit already, but you talked about your plan to open a data analytics unit in Clinton West. Can you expand a little bit on how that fits into Passage's strategy, and maybe discuss in a little bit more detail what kinds of analytics approaches you're focusing on, and for which kind of data? I know you mentioned next-gen sequencing as one. Are there other things that we should be thinking about?
Sure. I want to point out the analytical capability is a core into our drug development and how we advance these therapies from the research and from pen GTP all the way to the commercialization. To that end, we made a significant investment in 2021, and we opened our first analytical labs by the end of June of last year. Since that time, the team there has made an incredible progress, and we have been able not only to start some of these methods, not only to do tech transfer or develop some methods, but actually we have reached the point early this year to conduct a lot of product testing, supporting current and future manufacturing, but also we have started doing GMP testing. When you ask about the methods, we are looking into all the types of analytics that we are looking to characterize these vectors, both from a nucleic acid perspective and from a protein and from a biophysical perspective. Just to name a few, from the DNA payload perspective, we are testing, with a lot of the droplet PCRs, with qPCR, with NGS. When we are looking into the protein capsid, we try to characterize with capillary electrophoresis, also with mass specs. And then when we look at the biophysical characterization, such as full empty capsids, we are using AUC to characterize the level of full capsids, but also across orthogonal methods like SECMOLs and others. We are using also, interesting enough, a number of compendial, or not, I wouldn't say compendial, but more standardized methods, such as HPLC, ELISAs to characterize a lot of the impurities. For all of those methods that I mentioned, we are currently running them in our labs, and not only to, as I pointed out, to support our current and future manufacturing, but also to enable our process development and how we advance the methods, the production methods for our products to get to commercialization.
We thought that this was, Gil, I think we've talked about this before, but we thought this was a core competency we absolutely needed to develop. We think that the partnership with Catalent, based on ICM and based on the size of our population and the clinical study needs, that we can move forward with that externally. But we also knew, based on Alex's experience and our assessment of the field, that analytics and process development and ultimately product characterization from a quality perspective was getting rate limiting, cost limiting, and overall effectiveness limiting. So that's why we invested in this. We do have a lot of flexibility in this space as well to potentially build out additional capacity over time. So all of this is basically the core foundation of the overall CMC strategy that Alex is leading.
Right. That makes sense. And so this year, you've also done some reprioritization to extend your cash runway and focus on your lead clinical assets. So to what extent does the manufacturing strategy tie into that, or is impacted by those decisions?
Yeah, it's absolutely a key consideration. manufacturing and the investment in CM&C, and in particular the people and the strength that we've built around the competency and the depth of the group that Alex has brought together. So all of that is a key consideration for advancing the assets from early stage R&D to clinical development and future characterization. And because of the extensive R&D pipeline, we did have to earlier this year reprioritize our investments in both the future clinical development as certain assets and also the CM&C costs. And we've been partnering not only internally with Alex's group and thinking about how to optimize that, but also obviously with Jim Wilson and the GTP to think about that overall. As part of that, one of the core areas that we wanted to develop is not only some of the process development that Alex and his team are already working on, but we also thought about a pilot manufacturing suite so that we could actually bring that process development forward in potentially what could be actually a phase 1 suite over time. What we said is we wanted it operational by this year, and we've modified those expectations a little bit. So the full suite is not going to be operational by year end because we've modulated the investment there, but we certainly continue to work up our scale up capabilities. And so it's really to kind of modulate the amount of investments we're making and support the fully operational process and analytical development capabilities over time. But we thought that the pilot manufacturing suite itself didn't need to be built this year and it wouldn't affect our overall plans. So we've just modulated the timing of that, but some of the competencies are being built, just not in the suite, basically. So that was one, basically, adjustment that we've made.
Got it. So shifting to a different topic for the last few minutes, we've been hearing a lot about how the FDA and stakeholders in the gene therapy space have been focused on safety regarding AAV gene therapy. There was a panel convened last year. From your perspective, what were the takeaways regarding manufacturing from last year's panel and the broader kind of sense in the field, and what can be done moving forward to address some of the challenges?
Sure. So I think from an overall perspective, one of the themes that we saw going into this and actually coming out of it, and speaking with leaders in the field also, and regulatory leaders, as well as company people, as well as people like Jim, it was really about scarcity of data on many of these side effects that the FDA was concerned about, but didn't have enough either incidence or collection of the effect. One example of that is DRG toxicity, which is a theoretical impact, but is only the FDA panel only had 2 cases that were shown, one of which I think didn't have any clinical manifestations. So important to monitor, important to look at the potential outcomes for patients, but not really definitive guidance from the FDA at this point. So, and part of the idea was, can we collect more information? For example, on manufacturing, the primary concern was really regarding the fill and empty ratio and the potential impact on safety, for example, immunogenicity. Although, again, there's not a lot of evidence there. It's more to say, how do we focus on this? How do we collect more information? How do we evaluate this over time? And the main takeaway that we have is that, and certainly our focus, and certainly the link to the analytic build out, is the focus on quality and also control of the manufacturing process, and basically making sure that whatever we look at early continues to be evaluated over time and communicated and with FDA about any potential changes. So it's really about that quality metric and overall control, and that's what we took away from this. Not that there was any definitive negatives or positive. It was really about just gathering information, and we do support that and really think that that's critical to the overall safety of the field and evaluating the impact of gene therapy risk benefit.
Mm-hmm. Great. So in the last minute or so, I wanted to get your sense of, are there any CMC capabilities that you think investors or others may be underappreciating about Passage?
I would say, the fact that we have internalized a lot of these capabilities that you heard from Bruce and me earlier, and also the team that we have built. We have brought a great talent, a lot of expertise from the field, and we have been able to develop capabilities that they can really advance the pipeline, they can advance the company, and I would say in many cases also advance the field with some of the innovations we are making. So that would be my response to you on that one.
Yeah, and just to add, I think those strengths really underlie the continued build out of our internal capabilities that we think are critical to success, and that's why we're continuing to invest in Alex and his team and CMZ to drive forward the clinical programs.
Fantastic. So on that note, we're at the 25-minute mark. So I'd like to thank you guys again for your participation in our event today.
Thank you.